Hi, Dr. Hamish Fernando.
Hello, Young.
And hi to all our listeners of the BIOMEN Unreviewed, a podcast where Hamish and I, lecturers at the University of Sydney, chat about various aspects about biomedical engineering, but not necessarily in a factual manner, but more from our personal observations and experiences, and by no means peer-reviewed, because, well, who has time to peer review during a chat? Today we have a guest soon to be, Dr. Pooria Lesani, an expert in carbon dots for biomedical applications, and he'll join us today to talk about nanorobots and everything related. And I heard that you're going on a post-PhD holiday. So introduce yourself and let us know where you're going to, and so we could be all jealous.
Yeah. Hi, Young. Hi, Hamish. And hi, everyone. It's very good to be here. Thanks for having me and inviting me to this podcast. Yeah, so my name is Pooria. I just recently finished my PhD, in which I used to work on developing some kind of nanoparticles called carbon quantum dots. And I use them for so many different biological applications so far. And yeah, so I'm now doing a postdoc in the School of Biomedical Engineering. And yeah, in next 3 weeks, I'm going for a holiday. So
Where are you going? We could talk about the carbon quantum dots later, but where are you going in terms of holiday?
So first, I'm going to Iran to visit families. I mean, I'm having my sister and my mom here staying with me for last 6 months, but I'm going there to visit my dad and also my friends. And then for a week or 10 days, we're going to Turkey to explore some cities in Turkey as well. And then again, come back to Iran. And then after that to Sydney. Yeah.
And for more work, more research work.
Yeah, for more research and hopefully some teaching.
Sounds amazing, man.
Yeah.
All right, so I'm going to start off with a joke, as we do for this podcast. To an optimist, a glass is half full. To a pessimist, a glass is half empty. To an engineer, the glass is twice as big as it needs to be. Right, so, of course, it's a joke about safety factors and how all engineers have to take that into consideration when they're designing or developing certain things. All right, so today we'll be talking about nanorobots predominantly, and then in the second part of the podcast, we'll be talking about organelles, which are, I guess, in some ways, a type of nanorobot, right?
Yeah.
And so let's talk about nanorobots, and I guess I want to start off by saying how the media, in my opinion, has been generally sensationalizing about the development of nanorobots, right? And currently, most of the developments are in nanoparticles, right, rather than nanorobots. But having said that, though, there are quite a lot of research going on with nanorobots in using proteins to develop nanorobots, right? And so I do want to start off by just talking about what nano is.
Yeah.
And it's how it's usually compared in, again, media, that nano, they compare it with the size of a human hair, right?
Yeah.
So human hair is generally in the microns range or sub-micron range in terms of the thickness. Is it in the micron range or sub-micron range? Do we know?
I would
It's
say it's
around
about 50,000, 50,000 microns, if I'm not mistaken. So definitely
50,000 microns? That's a bit too
Sorry,
much.
nano. Nano.
So 50
nanometers,
microns.
50
50 microns,
So,
okay.
it is in the micron range.
Yeah, so every
My
time
bad.
you read an article about nanoparticles, it's like, oh, these are the size of, like, you could fit 1,000 of this or 5,000 of this in the diameter of a human hair.
Yeah.
Yeah, and so with these nanorobots, I guess let's talk about nanoparticles first. And I know, Pooria, you're quite sort of well-versed in this field. And so with nanoparticles, they're nano-sized. And if we talk about nano-size, we go from the scale from kilo to deca and then down to milli and then micro and then nano, so 10 to the power of minus 9. And so with this nanoscale, what are some of the, let's say, challenges in trying to make nanoparticles and nanorobots in your sort of view or in your sort of understanding?
Yeah, so to just give a brief, as Young mentioned, about the nano or nanoscale. So imagine, we all know 1 meter is equal to 1 centimeter, 100 centimeter, and then again 1 meter is equal to 1 million micrometer, and 1 meter is equal to 1 billion nanometers. So if we take 1 billionth part of 1 meter, it's going to be only 1 nanometer. So this is the scale that we are talking about.
Which is extremely tiny, right?
Yeah, it's very, very small and tiny. So the nanoparticles are kind of particles which are like billion times smaller than what we are in terms of like size. Or another examples to compare the size with the nano is that the size of nanoparticles compared to orange is like the size of an orange compared to planet Earth. So this is how we define the nano or nanoparticle
Right.
size.
It's interesting how you're bringing all these comparisons up because it's really the only way that I guess the general public and the general sort of population, right, would be able to understand how small nanoscale is.
Yeah.
Right. Like,
I mean, even I don't know whether I would be a member. I guess I'm a member of the general public as well. I don't know. I'm an academic also, but even I would find it difficult. I mean, I understand how small it is, but I would find it difficult to really picture without a scale like that.
Yeah.
Because it's beyond I mean, I don't know if you can conceptualize
Yes.
how small that really is without really comparing it to something like that.
Yeah.
and the thing is, you can't even see it under light, right? Like, it's in
Yeah.
a typical if you try, even if you zoom in as much as you can with an optical lens, you
You-
won't be able to see these nanoparticles
Nothing.
because they're actually smaller than the wavelength of light.
Yeah,
Yeah.
that,
Right?
that is insane to think about.
And so the only
Yeah.
way to actually visualize these nanoparticles or nanorobots in particular, right, is through other imaging techniques like electron-based imaging techniques, or even imaging techniques at the atomic level like atomic force microscopy and the like. Okay, so yeah, it's interesting how it's something that we can't see, something that we can't conceptualize. Usually these nanoparticles or nanorobots are in sort of solution, right? Like in terms of like it's in a vial of liquid and there's dissolved in it, or they float inside it. And that's the only way we could actually physically manifest and appreciate the existence of these nanoparticles, right? If there's a change in color in the solution, or if there is let's say, if you reacted with something, you could see the reaction. And so that's the only way we could actually see in the general sense, the presence of these nanoparticles. So let's talk about, I do want to jump into the whole toxicity aspect of nanoparticles actually before we talk about the actual uses, because the actual uses of nanoparticles and nanorobots tend to be along the lines of cancer treatment or cancer detection, right? Early detection of cancer and early treatment of cancer as well. But let's talk about the toxicity, because it's the general acceptance of these nanoparticles being, let's say, even if they're slightly toxic, let's say, in the most general sense, we're willing to use them to treat cancer, right? And it's that we can use things that are slightly toxic to treat something that's very bad.
Yeah.
In this case, cancer.
Yeah.
All right. So, Pooria, I want to sort of hear about your thoughts on the general commentary and the literature around toxicity of nanoparticles in general. Like you could refer to various types of nanoparticles. For the listeners, there are many different types of nanoparticles out there, right? With metallic-based nanoparticles, polymeric-based nanoparticles, ceramic-based nanoparticles, and also now with more research into nanorobots, molecular-based nanoparticles. Right. So I want to hear your thoughts on the general toxicity and how researchers are approaching this particular aspect in nanoparticle research and nanorobot research.
Yeah. Yeah, so so we have different type of nanoparticles. So and then based on what we are using to make these nanoparticles, they can have different level of toxicity that we know at this moment. I think one of the best examples of this nanomedicine technology that being used is developing some of this recent COVID-19 vaccine that some of us have already get these vaccines. Like I think Pfizer, maybe they have used liposomes technology. So the liposome is a type of nanoparticles that they use kind of fat. They made up from the fat from our body, so they have very low toxicity. And as Young mentioned, there are different type of nanoparticles. We can use some organic materials to make nanoparticles out of them, or we can use some metal ions to make nanoparticles. So there are different things. But I think worldwide there is a general concern, or not concern. There are an ongoing research that we all want to know that what's the level of toxicity is in relate to the nanoparticles in the long term, in the short term, because, the whole nanomaterials or nanotechnology field is very recent, so it hasn't been developed completely, and it's just a matter of time. So during the time, we are trying to find out the level of toxicity of these nanoparticles when we have them in our body in the long term. So in terms of the application that we can get out of these nanoparticles, there are many different applications that we can use them. Of course, we have so many biological applications out of them, but they can also be used for energy applications. So in terms of biological applications that if I want to point to a few of them, they can be used for like biosensing and bioimaging. So what does it mean? So like in terms of bioimaging, sometimes we know that something is wrong in our body, and then basically most of the time we need to do a surgery, open up our body, and then see what's happening inside. But some of these nanoparticles that we are making, hopefully if everything works properly, we can just easily inject them into the bloodstream in our body and then help them to go to the targeted area. And then using some advanced microscopy, we can visualize inside our body like non-invasively without opening up our body. So this is kind of bioimaging application out of them. And also for biosensing, like let's say like Alzheimer's disease or Parkinson's disease, kind of we know some kind of treatments at the moment for this disease, but the problem is that we don't have the technique for early detection of this disease. So when we find out like we have Parkinson's, it's too late, So again, using these nanoparticles, we can improve this early diagnosis of disease. So we can find if anything is wrong in our cells at the very, very early stages, and then we can immediately start the treatment, and hopefully we don't let the like a disease to progress in our body.
Right.
Yeah. So and another like very, very attractive kind of application out of these nanoparticles is the targeted drug delivery application. So many researchers in the world are now trying to make different type of nanoparticles and use them for different targeted drug delivery application for cancer therapy and so many other similar diseases that we have, and we are very limited with the treatments. So I have a very nice example again for how these nanoparticles would work for the targeted drug delivery. So it's pretty similar to when we want to go somewhere. So we have two options, like at least two options. We can catch an Uber or we can use the public transport, right? So this is the same that when we have a headache, let's say, when we take a painkiller, this painkiller will be distributed all over our body, and some small part of that painkiller will be taken up by the area that we have pain. So this painkiller goes so many places that we don't even want it to go, right? So imagine you want to go somewhere now, and you have those two options: the public transport and catch a Uber. So when you get a bus or something, so the bus will go all over the city and it stops at different bus stations, and so you waste so many times and stuff.
Right, so you go to places where you don't want to go. Like
exactly.
With public transport, because the bus stops basically everywhere along its route.
Yeah, so for example, I'm coming from Five Dock to Sydney Uni. So if I want to catch a bus, so it goes to Leichhardt, it goes to Petersham. There's so many suburbs, and then finally I get to Uni. But as Young said, it goes to so many places that I don't want to go. But like imagine you catch an Uber. Okay, so the Uber will act as the nanoparticles, and then the passenger the passenger that sits in the Uber is the drug. So that's the drug that we can load to the surface or to the structure of the nanoparticles that we have.
Right.
And also we can load some specific materials on the surface of these nanoparticles that we call it targeting ligands, that they are specified to go and attach to a specific type of cancer cells or any cell types. And this is the Google map, or this is the GPS, right? So we have the Uber, which is the nanoparticles, we have the drug, which is the passenger, and also we have the targeting ligands, which is the Google map. So we set all of these three items together, and then we inject it to the a bloodstream. They start going all around the body, but because we already gave them the direction with the targeting ligands, they will only go to, let's say some person has skin cancers or breast cancer. They will try to directly go there. As soon as they get there, they stop, and then they ask the passenger, which is drug, to get out of the car. And then hopefully they will, by releasing the drug, they have like high efficiency treatments
Mm-hmm.
only on the cancer area, not on the healthy cells. Yeah.
It's a nice analogy, Pooria.
Yeah.
Do you use that in your class as well when you're teaching?
Yeah, sometimes, yeah.
Yeah.
So, like when I want to explain about the applications of this material, so it's always good to be very simple. You know?
Yeah, absolutely. That's awesome. And one of my questions was how, when you mentioned the bioimaging, and I was going to ask you how you managed to get the nanoparticles to the target area. And now you've explained that by talking about the ligands.
Yeah, exactly. So
Yeah.
that's one way that we can guide them to the area that we want them to be. And also the other thing is that like we have some type of nanoparticles that they have magnetic properties. So even with the external magnet, as soon as these magnetic nanoparticles are in the body, by an external magnet, we can somehow also
That's really interesting.
Like
Yeah.
remote control,
Remote control, yeah.
control magnetic nanoparticles.
yeah, exactly.
Right, so if you put a magnet near your head, then all the magnetic nanoparticles that have been injected into the body go into your brain
Yes.
or through the blood vessels.
Exactly. Yeah.
Is that done now?
It's something that, as also I think Young mentioned, it's highly it's been.
There's a lot of
researching
research
now.
going into those kinds of nanoparticles where you've got magnetic nanoparticles. Of course, magnetic nanoparticles have magnetic properties. You put them into the bloodstream, right, and then use a piece of magnet to get them to a cancer or to a particular tissue.
Oh, that's really cool.
And also with the fluctuate. Okay, so something about with magnetic nanoparticles is that within a fluctuating magnetic field, they generate heat, right? And so you could also fry the nearby tissue around the magnetic nanoparticles. Of course, you don't want those tissues that are fried to be healthy tissue, right?
Yeah.
But if for cancer tissue, right, frying them, and they're more sensitive to heat generally as well, then you kill the cancer, or that you destroy the tumor tissue at least, right? And so there's a lot of research going to
That's been performed experimentally.
yes,
as
it's
well.
been performed experimentally in animals. Yeah.
Right.
Yeah, you know, our normal body temperature is 37 degree, right? So by having this type of magnetic nanoparticles or any type of nanoparticles that can produce heat, we can send them to the area, like let's say a cancer tissue in our body. We can guide them to directly go there, and then when they are there, we expose them to a strong magnetic field, and then the localized temperature will be increased up to 45 or 50 degree. Only in that cancer area. And then because you're much higher than the normal body temperature, as Young mentioned, they just fry the tissue and then they kill the cancer tissue.
So just from a more layman's
Yep,
perspective, because I'm not an engineer like you guys, so when you create that heat, how about, wouldn't the heat dissipate? Or is that so, like, it's such a pinpoint area that there's not going to be any harm to surrounding cells, is it?
Yeah, so in the ideal situation, all of these nanoparticles will be uptaken or will go to the cancer area only.
Right.
And then when they start producing heat, it's going to be like for 10 seconds or 15 seconds. So if if they start producing heat for a longer time, definitely they will affect the healthy tissue around it.
So one of the challenges would be to engineer such a way that you control the amount of heat dissipated or a controlled duration so that, of course, the surrounding healthy cells and tissues may or may not be affected by the heat generated.
Okay.
But the idea is to try to heat up the cancer cells and the cancer tissue predominantly.
Right. And like you said, the cancer cells are more sensitive to heat. Generally.
They are generally more sensitive to heat.
Okay. Okay. With regard to the targeted delivery, now you mentioned liposomes being used for the vaccines, correct? Is that I guess it's not highly targeted, but does that contribute to the targeting, or does it just only allow the uptake into the cells? Does it make it? I know it should make it easier, but is the purpose of that just the easy uptake into cells and through membranes? Is that why they're using the liposomes?
Okay. So the question is that why they chose a liposome.
The liposome specifically.
Liposome is one type of nanoparticles that is being researched and developed for like a long time.
Right.
And because they are just made from some biological components, they have a very, very low toxicity or even no toxicity because they are just the components is some part of fats or something that we can also find in our body.
Right.
So the main reason that they chose those type of nanoparticles for developing vaccine is that because in regards to other recent developed nanoparticles, we are just able to go up to the like animal studies, trying them in mice or some type of animals, but nothing more than that, because we are still not sure what's going to happen to them in the long term.
Yeah.
So when we have liposomes in the body, after a while they start
Metabolizing.
metabolizing is
They're
happening,
metabolized.
and then they will go away, because we know that they're gonna be digested by the body, and then they will just release through urine or something.
Yeah.
But
So some of the listeners might be sort of aware of the fact that cell membranes are made up of the lipids that you mentioned, typically glycerophospholipids, and with these membranes, right? And liposomes also made up of these glycerophospholipid bilayer membranes. And some of you might think, do these two membranes fuse together for the nanoparticle to work? And surprisingly, at least from my perspective, that's not the case, right? The fusion actually is very rare, right? It's usually endocytosis, right, where the cell brings in the liposome as a whole into its own vesicle into the cell, right? And then the metabolistic processes take place. So the lysosome pathway, where it starts to degrade the liposome, and then it releases the content inside the liposome, right?
Right.
Yeah, so that's how these nanoparticles generally tend to work when they are designed to go inside a cell to do its function, right? Whether it's to deliver a cargo or deliver a drug or deliver mRNA, DNA, or to become a beacon to help with imaging, right? So with carbon quantum dots, and quantum dots in general, and other gold nanoparticles or magnetic nanoparticles, they get endocytosed inside the cells,
Right.
right? And then they do their sort of shining, they do their color there, rather than being fused with the cell.
Right.
Yeah.
Going back to your comment about the recent vaccines for COVID being lipid nanoparticles, so liposomes, I wanted to check if that was indeed the case, and that is indeed the case. And what that reminds me right now is that these liposomes, right, one, they're, as you mentioned, they're made up of bio-based materials, and so they actually have very little toxicity, if any toxicity, and they'll be metabolized by the human body, right, so the cells and the biomachinery there. And what also reminds me is that these nanoparticles, liposomes and the like, they're there to really protect the actual thing inside, right? So in this case, with the mRNA vaccines, they're there to protect the mRNA, right? Which is then the activator or the thing that does its job in terms of providing the immunity against, let's say, COVID and other virus-related diseases, right? And so you also mentioned about the targeting aspects, and what I wanted to sort of bring up there is, again, if you did the sort of high school biology or even high school science, we usually talk about the lock and key model, right? Where two enzymes or two proteins typically exist in a way that they match, right? And so if you know what protein you're targeting or what molecule you're targeting inside the body, you could actually design the nanoparticle in such a way that you could have some of these proteins on the surface of these nanoparticles to bind essentially via the lock and key mechanism to target certain areas of the body.
Right. And that's what you mentioned. You were talking about ligands earlier.
Yes, and that's the ligands.
Mechanism of it,
yeah.
right?
Yeah. Cool. That's so. So we've been talking mainly about nanoparticles, and we've sort of brought up liposomes. We've also mentioned in passing about gold nanoparticles, magnetic nanoparticles, and both of them, both gold and magnetic nanoparticles, have the capacity to heat up, right? If you, for magnetic nanoparticles, if you apply fluctuating magnetic field, magnetic nanoparticles heat up. For gold nanoparticles, if you fire some infrared light or near-infrared light to where the gold nanoparticle is, the gold nanoparticle heats up. Right, so they're interesting mechanisms, right, in terms of trying to kill cancer cells, trying to kill cancer tissue, and going back to the whole application of this then, right, with treatment of cancer, right, it impacts so many people around the world, right, and there's so many different types of cancers, and with these nanoparticles, right, I presume then, right, there will have to be multiple designs of these nanoparticles, right, and one nanoparticle design wouldn't be suitable for all types of cancer, right? So it's different cancer types would necessitate different nanoparticle design. Do you agree with that sentiment?
Yeah, of course. Yeah. So, for each type of cancer, so we can use like let's say the iron oxide nanoparticles can be used to treat different type of cancers, but what we need to do is that to design based on the cancer or the area that we want to aim. We need to design these nanoparticles. We need to engineer these nanoparticles in terms of size, in terms of what we should have on the surface, in terms of what should be the surface charge of nanoparticles. So I'll give you an example. So,
And also the mechanism of killing, right?
yeah, mechanism of killing. Do we want the nanoparticles to only deliver the drug to the area, or we want the nanoparticles to deliver the drug and also produce heat? And like based on what we are thinking of the treatment. So the design can be very, very different based on the application actually. So let's say we want to treat a brain tumor using nanoparticles. So this is one of the most challenging application that we can get out of the nanoparticles to treat cancer. And then the main reason is that the nanoparticles, since they are injected into the blood, they need to pass blood-brain barrier, which it's the very, very strong filter under the way.
Like you shall not pass kind of barrier.
Yeah, exactly. Yeah, yeah. So you need to make sure. From what I'm understanding from this application is that you need to make sure your nanoparticle size is less than 5 nanometers, which is very, very small. Like the average particle size of gold nanoparticles or iron oxide nanoparticles is something around 20 or 30 nanometers. So if you want to use them, you need to find a way to reduce the size below 5 nanometers. You need to somehow have a surface charge, like because this, like imagine a spherical thing that's the nanoparticles. They have a surface charge, so you need to have a kind of neutral or positive surface charge of these nanoparticles, because from the research it was found out that if the surface charge is kind of positive or neutral, it's easier for the nanoparticles to pass the blood-brain barrier.
Right.
Yeah, and also you need to again think about the hydrophilicity or hydrophobicity of these nanoparticles. So there are so many things that you need to consider in the design, just the nanoparticles itself. So imagine you designed and engineered everything about your nanoparticles that can easily pass the blood-brain barriers. So the next step is that to find the best way to treat that cancer or the brain tumor. So is it just delivering one drug is enough? Or you need multiple drugs, because some of the tumors, they are kind of developing the multi-drug resistance. So even if you deliver tons of drug to that area, they won't let the drug to go inside, because there is a mechanism happening in some of the tumors, the multi-drug resistance, that they are very resistant to uptaking the drugs. So in some cases, then you need to, while the nanoparticle is going there, not only, the nanoparticles should carry 2 different type of drugs to have like a efficient treatment in the brain tumor. So that's another thing that you need to consider. And also, based on the area of the brain that this nanoparticles is going, you need to have that like type of biomolecules or targeting ligands that can help the nanoparticles to directly go to that area, not any other places in the brain, because brain has a very complex structure.
Yeah. Of course. You want the brain to have a complex structure, otherwise we wouldn't be able to do the stuff that we do.
Yeah.
And just a quick heads up for the listeners who might not know what the blood-brain barrier is, just so that you know, the brain is extremely sensitive to external fluctuations. So the brain has a very tight barrier in which it restricts the types of molecules or different ions that can move in and out.
Again, you shall not pass.
Yeah. So therefore, that's why Pooria was mentioning just now, delivering drugs to that area becomes really difficult, especially in the brain, because of that barrier.
Okay.
I wanted to extend on what you were talking about. You mentioned that the tumors, they become resistant to the drugs that we're sending in, even if it's a multi-drug therapy, right?
So sometimes, yeah. Sometimes, when you deliver some specific type of drugs to this cancer or to these tumors, after a while they're getting a bit resistant to that drug. So
they're adapting to the drugs. It's like a little mini evolution or something going on.
Yeah, yeah. I don't know. Something happened inside.
So what I suspect is happening is that, like, once you start the treatment, most cancer cells will die. Some of them may or may not live, right? And those that do live, probably they have some sort of genetic mutation or genetic sort of characteristic that makes them, by chance, resistant to these drugs. And when they multiply, right? Again, it's a game of probabilities there,
right?
Where now it's highly likely that most of those cells from the tumor cell that was essentially multiplied from, right, that happened to be resistant to that drug. Now most of the cells are now resistant to that drug because they've been duplicating from that.
Yeah.
Right? And so in the longer term, I guess it develops that resistance.
I find that really interesting because it's like antibiotic resistance.
Yeah.
But in the case of antibiotic resistance, the reason it's so widespread is because the bacteria can tend to actually spread, but the tumors, I assume, would not spread from person to person. So even in that small stage within one person's lifetime, in that small time period, even with multiple drugs, that it can become resistant.
So
That's amazing.
I guess it's a terrible luck of it's a terrible lottery sort of thing.
Yeah.
When it comes to that, so there's other chemotherapeutic drugs which are sort of universally killing, that kills basically healthy and cancer cells, and I would presume then for those kinds of, let's say, drugs, even those kinds of drugs, the cells might develop a resistance to it eventually, right? Like at an individual level, right? Not as if like with bacteria where it spreads.
Yeah.
Right. Just before we move on to our next topic, I do want to just go back on the whole aspect of targeted drug delivery, right? And why that's important for us to really, if we have like a toxic drug that's designed to kill cancer cells but and also be able to generate side effects on the healthy cells, why that targeted is important. And you mentioned the whole sort of analogy with Uber and the Google Maps being the targeting ligand, let's say, to help the Uber driver get to where they need to go, with the passenger being the drug to kill the cancer. All right, so it's important because, again, if these drugs that are inherently toxic, right, they're designed to kill cells, right, mostly cancer cells, but also it damages the healthy cells too. Right? If they spread around the body, then you get side effects, right? That's why people lose hair if they go through chemotherapy, because one of the effects of some of these drugs is the alopecia, or the loss of hair, right? And also other side effects like nausea, headaches, and the like, right? And so you've got these side effects, and you want to minimize those side effects, right? And so that's why we want to just deliver these toxic drugs to where we want them to go to, right? And so that's the importance of the targeted drug delivery. So we've been talking about nanoparticles mostly, right? So, and they're just, to me, what they are just simply vesicles, or they're simply, again, cars or payloads or vehicles to transport the drug or to transport the fluorescent molecule for diagnosis and treatment. There's also, I alluded to this at the beginning of the podcast, where we have now research into nanorobots, which can actually do stuff. Like, instead of just delivering, right? Instead of just being a delivery truck, right? They are machines that that can actually move, right? And that can actually control certain physical aspects of biological function, right? And so, and these we call them nanomachines and nanomotors, right? And most of them are actually nanomotors. They rely on the interaction between the protein chains and the protein domains, and hydrophilic interactions between the protein sequences. So if going back one step up, proteins are made up of sequence of amino acids. Various amino acids have various properties. Predominantly, one of the main properties is its hydrophilicity and hydrophobicity, attraction and repulsion to water, right? And depending how close these amino acid sequences are to each other, right, and if they're in some sort of aqueous environment, they interact with each other to essentially behave like flagellation or motors, right? And it propels and applies force in a particular distance, or towards a particular distance or particular direction. And researchers have essentially encapsulated this and taken this to develop nanomolecules, right, and synthetic nanomolecules, nanomotors and nanomachines, in a way that they can control how much force is going into one direction and how much, let's say, force is applied, how much velocity is applied into this to create these machines that can actually physically move around. And what I was thinking is that these nanomachines and nanomotors that we're synthetically developing right now, they're quite similar in its function, where it does involve the interaction between these protein chains. It's very similar to how our natural organelles work, right, inside the cell, right? An example I present to you here is like the ribosome, right? Ribosome is a protein. Well, it's a group of proteins where they bring amino acids from the cytoplasm, right, and connect it to the mRNA that's passing through it, and it lets mRNA pass through the ribosome, attaches an amino acid that's matches the codons, or the three sets of amino acid sequences, right? And then it moves the mRNA along and then brings the amino acids and builds up an amino acid to create another protein. That's essentially a nanorobot, by definition, right? And so I want to now talk about organelles, right, in response to the fact that these nanomachines and nanorobots are essentially what we're trying to do is create new organelles, really, right? Whether it's outside the cell or inside the cell. And so I'd like to sort of get your thoughts on what you think about organelles and sort of what you appreciate about these organelles inside the cell. And I might ask Hamish to sort of talk about organelles.
Right. So with regard to the ribosomes you were talking about, something I talk about with my students quite often is we talk about how genes and how our DNA makes us who we are. And I sometimes ask them, so how exactly does this DNA or your genes make you who you are? What is it about the genes that make you unique? And the answer is basically DNA has genes code for proteins, and proteins structurally and functionally to a great extent make you unique. All right, your hair color, your eye color, skin color, all that, you know. So when you're looking at how your DNA ultimately, becomes proteins, that whole process is an absolutely intricate, beautiful, amazing molecular process and involving all sorts of, like you mentioned, essentially nanorobots, molecular machines. So just going through it, when you think about just once your genes first split up and you form the messenger RNA. So to move from DNA to proteins, you got your genes. Your genes need to form mRNA through a transcription process. Then
that happens in the nucleus,
Yeah, that happens in the nucleus. The mRNA leaves the nucleus, and then ribosomes bind to that, and through a translation process forms your amino acid chain, all right, which ultimately gets processed and folds up into your ultimate protein. Now, that's quite a complex process, but just imagine in your body that you already have, just through, of course, millions of years of evolution, that you have these little nanorobots capable of taking a, double-stranded DNA, splitting it up, binding, more nucleotides to it to form an RNA strand. We can't, with all our knowledge right now, we can't perfectly replicate that.
But we're trying to.
We're trying to.
Not trying to replicate it, but trying to develop similar machines that could do other biological functions.
Exactly, yeah. And like I mentioned in the last podcast, I do believe, I have a lot of belief in human intellect, and I do believe that one day we'll do that. But this is, when you think about evolution, all right, this whole process happened through a series of trial and error, and ultimately we got this process in, which, just... to me, it's absolutely mind-boggling how this can happen, because obviously the first life form, let's say the first single cell, didn't have such a complex process, and just of course through millions of years of trial and error, we ended up getting this. So, like I could go into the intricacies of how this happens, but before I do, I just want to take Pooria's take on this as well, as somebody who works in nanotechnology as well, about.
I'd like to frame that sort of Pooria's, let's say, opinions on how close are we, or how far are we in terms of trying to replicate our cellular machinery, or what cellular machinery can do, right, with synthetically developed nanomolecules or extrinsically developed nanomolecules and nanorobots?
I think, like, based on what is happening all around the world these days, and the interest that the researchers and experts like showed with this field, or the field of like nanorobots, or in general nanomedicine, I don't think it's too far. It can be very close. Like,
So my understanding right now is with these nanorobots, they can move from point A to point B because they have the motor, right, which is made up of proteins that can sort of revolve around, and it's similar to how a propeller in a boat runs. Right? So there's been more developments than that.
Yeah, I think so. Yeah, and then I think there are so many like recent developments, like in laboratory developments, now they are trying to try it in the like a animal, like perform some animal studies and then see how it goes in there, and then which is already done as well. But then the next step would be the like trying on humans, hopefully without any toxicity or any side effects, and then if it all works, then it's gonna just like change the whole treatments all around the world, you know, because it's gonna be very accurate with high accuracy. So we can ask these nanorobots to go from point A to B and do this for us and then come back. So it's very straightforward procedures for asking these robots to do in our body for different.
So with these nanorobots, I guess the main aim for these nanorobots to be doing, right, aside from moving from point A to point B, which by itself is, I think, useless, right? Yeah. It's to facilitate the protein reactions, right, essentially, right? So to control certain reactions within the biomachinery and essentially be an extra organelle, right, to do certain things. Is that commentary somewhat correct?
Yeah, I think so. Yeah, so from what I know about this nanorobots technology, so that can be completely right. So I remember there is a paper published, I don't know, a few months ago in Nature that they are also discussing not about like a research results or something, but about the future of these nanorobots in the human health and about what they can do in the body, which was very, very interesting. And everyone are very like optimistic about the future of these nanorobots. So let's see.
Yeah. So just to reiterate, with these nanorobots and nanomotors and nanomachines, I think I mentioned this in just a moment ago as well. It's that they rely on the protein domain dynamics. Right, so how the proteins interact with each other, how the molecules interact with each other, at essentially looking at the types of bonds and types of interactions, hydrophilic, hydrophobic interactions, van der Waals interactions, secondary bond interactions between various sequences within the protein, right? And so what that also reminds me is how our actin and myosin filaments in the muscle work, right? The way that they contract, right, is obviously there's some pre-signaling that happens, right? That and then that triggers the actin. Is it the actin that looks like the bean sprout, or is it the myosin that looks like the bean sprout?
The myosin is a thicker one that's actually pulling on the actin. The actin is the ones that twist around each other.
When there's interaction between the actin and myosin, the actin head bends,
the myosin head binds to the actin and then cocks.
So the myosin's the bean sprout one, and then
the myosin has the head which pulls on the actin.
Yeah, so actin is the filament that's within the cytoplasm. Yeah. And so you've got the myosin that bends, and then that creates the contraction.
Yeah,
right?
exactly.
And if you've got many of these actin myosin interactions, then you could actually see a physical manifestation of the muscle contraction.
Muscle shortening, yeah.
Right, so essentially we're taking that principle, right, with these nanomotors and nanomachines, right? How these proteins interact and apply force in a particular direction or move in a particular direction, right? And also how it facilitates certain protein reactions, right? That happens in the biomachinery, which I think is quite fascinating. And to be able to do that and be able to control synthetically these molecules at the molecular level, right? We're at the stage of technology we can actually do that in, right? And so it's something that we can really look forward to in terms of the development in this space. Yeah.
So just expanding on this, it's just a question I normally deal with recently. So as of recent, I've always been someone who, when I think about the aging process, I am someone who is horrified of aging and, just Not to get all deep right now, but just in general, okay? But just to ask the two of you, if you could live for, say, 200 or 300 years,
Yeah.
would you?
Yes.
Pooria?
Yeah, if I can be
Yeah.
like healthy.
Healthy,
Yeah,
healthy,
of course.
healthy living.
Yeah.
All right. Okay. So same here. So what I
Okay.
was thinking was, now, when we age, the reason we age primarily is because a lot of the molecular machinery, one of the major reasons is because a lot of the molecular machinery start becoming dysfunctional.
Okay, I see where this is going.
Yeah.
Yeah.
So, for example, when we're talking about the DNA replication process, okay, normally there are so many checks and balances, because every time your DNA replicates, there's going to be an error somewhere. But your body has other machines that come in, checks, double checks, triple checks, quadruple checks to make sure everything's perfect. As we age, the machinery becomes dysfunctional. Okay, so that checking process is now lost. The quality assurance is lost, and therefore you come up with cells, or DNA is produced, and ultimately cells, tissues are produced, which aren't as, say, to put it mildly, high quality as it was when you were younger. And that is a major reason we go through the aging process.
But you lose fidelity, essentially.
Exactly, yeah. So, another thing that might be really cool, hopefully, and something that I would be really interested in, is maybe down the line, once these start becoming dysfunctional,
You can replace them. Yeah, you can replace them with nanorobots that
just
can
inject some of those nanorobots and let those
That's
bad
it.
boys do their job.
Yeah, I think as you mentioned, everything is started at the molecular level. So as soon as we have the technology that we can like make the dysfunction biomolecules to work again, or the cells to work again, so that's it. I think not only aging, maybe other like type of diseases also can be like solved. using this
Diseases for
technology.
sure, yeah. But here's the beautiful thing. If you think about it, a lot of the diseases we get as we age is because we're aging. How many people do you,
Yeah,
I
that's
mean,
right.
yeah, cancer, diabetes, lots of cardiovascular diseases.
stupid thermodynamics.
Yeah. It's a result of aging,
Right.
unless it's an infection. And even in infection, you're able to fight it off better generally when you're younger, because your thymus and your immune system as a whole functions better.
stronger,
And that's also a result, as you age, everything just becomes more dysfunctional. And I've been reading a lot on aging as of recently, and just if you think about it, right now in medicine, we're doing this specified approach where we're trying to fight cancer, we're trying to fight cardiovascular diseases, we're trying to fight diabetes, all sorts of chronic diseases. But one of the major root causes of all of them is simply the dysfunction that happens as you get older and older. Because the probability you'll get any of these diseases is greater when you get older. So if you can stop, or if you can minimize the damage that aging causes, that in itself would feed back on every other disease that you're seeing, and it would reduce the extent of that happening.
Yeah,
I would imagine. So,
Yeah.
Right.
So how long will it take for these nanomachines and nanorobots to replace all the damaged machinery in our cells? And
Pooria,
I
save
think
us.
based on
Pooria.
what Hamish mentioned, we need to work harder from today.
no, so I'd like to really, I bring this up in my lectures as well, but I really like to think of cells as machines, and sort of relating to this. All right, cells as machines, as I say, with cells, there's an input to the cells, and depending on that input and the environment around the cell, it generates an output, right? Where those outputs tend to be sort of tissue, proteins, and if the cell machine is working well, then it produces healthy outputs, right? If the cell machine isn't working well, it produces unhealthy outputs. Again, I'm overgeneralizing here, but I hope you get my point. If we can now use these nanorobots to replace the broken parts of the cell machinery, then instead of these old cells or sort of otherwise unhealthy cells to be producing unhealthy products, we can add nanorobots and nanomolecules in there to have them make healthy products, and hopefully that will result in 200, 300 year lifespan.
I guess that would be awesome. Part of the reason I want to live for so long is because there are all these new discoveries that are coming up, and I want to be there when they're discovered. I want to just experience
Very
that.
exciting. Yeah.
Like, can you imagine, like, if they come up with a solution for like the theory of everything or something, and I happen to pass away 2 days before that, I'd be like, no, I wanted to be there. Like, just my fascination for all of this. I want to be around when all these things are discovered.
I would be surprised if it was just a single event, though. It would be like the build-up of these researchers and build-up of like findings to end up being something that's clinically applicable, right?
Yeah.
Like it's not gonna be like one research group's like, hey, we found this.
Exactly, and which is why you said that the movement or the walking, was that what you said? The nanorobots are capable of movement?
Yeah, it's moving from point A to point B. Essentially a lot of actin filaments. Right?
Yeah, while that in itself is not going to do much, that's a start. So
yes, indeed.
Yeah, that's the first step to getting something else done.
Indeed.
But like for developing the COVID-19 vaccine, like we can say like they were working together to develop this vaccine. So if we do the same thing with like developing this like nanorobots or developing nanomaterials for specific applications, if all like research, like large number of researchers start working together instead of kind of competing or something like that, then we can get to this stage that have them like at least into the clinical trial very, very soon in next, I don't know, few years. A COVID-19 vaccine was developed in less than a year or maximum 1 year. So because so many people
collaborating.
Collaboration, you know. I mean,
apparently the actual science, like the actual product was actually developed very, very quickly, even sooner. It's the regulations that
yes,
that took
exactly.
the time, right?
Yeah.
Yeah, to make sure that it was working well.
In few months they could like at least have some very good experimental results in the lab, and then the trials, and then up to 1 year we had it in the market, and then we were just getting vaccination to be to protect ourselves against COVID-19. Like I think it's the same story with other technologies like the nanorobots. So if the collaboration happens all around the world together, then we might have it soon, and none of us will get old.
I have a quick question for you before we wrap up, right? With these nanorobots and nanomachines, we've been talking about how they're made up of proteins, synthetic biomolecules. Do you think it's possible to actually make these nanorobots with synthetic materials? Not let's say proteins per se, but like with, let's say, gold or iron oxide particles or liposomes. Are we able to make nanorobots with them, or are we sort of limited to the domain of proteins where you do have those sort of hydrophilic interactions that allow them to move and apply force?
No, I don't. I think we don't have really limitations of what kind of nanoparticles that we can use to developing these nanorobots. I think there was a research group trying to make nanorobots out of the iron oxide nanoparticles. It's just a matter of how you're gonna engineer and design the surface.
And you have to get it down to pretty damn small as well, right?
Yeah,
Molecules are sort of inherently small. They're like less than tens of nanometers inherently, right? Like it's with the synthetic materials, there's also manufacturing challenges where you do have to have these manufacturing techniques to have it go below a certain resolution, right, in order to make those little parts.
Yeah, and then the other maybe challenge that we didn't talk is that, okay, so you made everything works perfectly fine in the lab, and then if you inject it to the animals or to the body,
the body's a complex thing.
Yeah, exactly. As soon as they are in the bloodstream, the body, they might recognize this whole nanorobot or nanoparticle.
Stupid macrophages.
As a foreign materials. And then that's their job, to destroy it. They will attack to the whole system that you spent 2 years or 10 years to develop. And then
actually, that's actually a pretty big area we haven't even talked about at all yet with the macrophages. Like if you add anything foreign, like nano or whatever, right, they'll just start attacking it. And so you do have to make sure that the macrophages don't destroy everything before it does anything. Yeah,
it's again, there are so many factors that can affect that you can reduce the risk of macrophages attack to your system. Then you need to consider in your design as well.
Right.
So what I'm really hoping for is that one day we see so many of these nanomaterials that develop to get into the stage that we can use it in the market. I mean, we can see now the quantum dots in the TVs, like QLED TVs. So this is one application, not in bio, but hopefully one day we can see it.
Well, there are again, like COVID vaccine as an example. There are nanoparticle-based clinical solutions that are in the clinic right now. Right? Another one is Doxil, which for those that are sort of in the nanomaterials field would be able to know what it is. It's a chemotherapy drug that's been coated with a liposome for cancer treatment.
Right. I think we have some nanoparticles in cosmetic applications.
Yeah, and there's nanoparticles in cosmetic applications, and we could talk about this all day. I just wanted to just bring up also while we were talking about organelles, like, can we just talk about how the names of those organelles are wonderfully named, like endoplasmic reticulum. It sounds so good. Mitochondria, the powerhouse of the cell, right? And the Golgi apparatus. Like, I think they're named fantastically well, and I think hopefully with the synthetic nanomolecules that are coming through, hopefully we can replicate the complexity of those wonderfully named organelles.
Hopefully one day. Yeah.
Yeah.
And just off topic, speaking of funny names, like I've always found the funniest names to be in when you're talking about cosmology and astronomy. When you Big Bang, Big Rip, like it's so basic with the way everything's named. Milky Way, like everything's named just in such a generic, like whatever name. Whereas when you go to biology, you got naming muscles and bones and all that stuff.
All Latin,
Yeah, it's all Latinized and complex, you know. Whereas, I mean, think about it. What, like
cosmic background radiation,
inflation,
That's right.
black hole. I mean, neutron star. I mean, it's just so basic. It's not difficult to remember at all. So whoever studies that, I mean, they go through probably a lot of complex math and all that, but at least the names are easy to remember.
But that's a big challenge, actually, to remember the names sometimes.
In biology,
Confused.
remembering the names is
It's
tough.
a lot. It's a lot.
Speaking of challenges, it doesn't look like Manchester United's going to be in the Champions League anytime
Oh
soon.
my God.
For those that are listening, Pooria is a big fan of Manchester United. And also as disclosure, as I'm a big fan of Tottenham Hotspur, and they won the two big games recently against Arsenal and against Burnley yesterday. Manchester United hasn't been doing that great though. Oh, they beat Leicester 5-1. I think, did they beat Leicester 5-1? Let me check.
No, I don't think so.
Was that Man City? I can't remember. What? Anyway, Manchester United's in a big sort of
So it doesn't change. It doesn't affect anything. I've been like following Manchester United for I don't know, since for last like 15 or no more 20 years. So since I was 11,
hardcore fan.
Yeah. So yeah, I'm a very like huge fan of Man United. So Wayne Rooney is my hero.
You could tell Pooria's age by him like mentioning Wayne Rooney as the main star.
That's right. Yeah. And then so
not old enough to follow Beckham, but old enough to follow Rooney.
Yeah. So like since Sir Alex Ferguson left Man United, so it's a big challenges that all the club is facing, and then they're trying different managers. So hopefully this year when Mr. Ten Hag comes to the, I think he's coming to the club today. So it's the 16th of May. So hopefully he can bring some good players and get rid of.
Mourinho should have come straight after Sir Alex Ferguson.
Yeah, that was
instead of David Moyes.
Or Guardiola was the best options after.
I don't like Guardiola. He just follows good teams, and I don't like him. I don't know. He seems too arrogant for my liking. Even more arrogant than Mourinho. I don't know.
Yeah,
I'm a big fan of Mourinho. He shouldn't have been sacked by Tottenham Hotspurs either. But that's a very unpopular opinion. If I say that on Reddit, I'll be downvoted a lot.
But anyway, I'm pretty sure Man United will back to the first stage very, very soon. So with the new manager, new good players, and we have Ronaldo in the front.
He's getting old. I'm a big fan of Ronaldo. I think he's better than Messi. Again, I'll be downloaded for heaps if I say that on a forum. But he's getting old though, and then hopefully maybe he might be like LeBron. Like LeBron's like almost 30. What now? 30? How old is he?
He is 36 or 37.
Oh, he's actually younger than Cristiano Ronaldo then.
Yeah,
Ronaldo's like 37.
Yeah, but I mean,
let me check.
Yeah, Ronaldo's
LeBron.
LeBron's a freak of nature.
Oh, he
My
is.
goodness.
LeBron is 1984 December 30.
So 38.
38.
37.
37. December 30. Yeah, 37.
And Cristiano Ronaldo. 1985. So LeBron's actually older.
Oh,
Yeah.
LeBron's insane.
Cristiano Ronaldo's 1985 February 5. So 2 months apart, or around 2 months apart.
Yeah.
LeBron's old. Oh man, what LeBron does though, like, is in terms of like jumping around like that.
crazy.
Speaking of basketball as well, I know we've sort of slightly gone over time, but that's fine. You guys can stop listening if you don't want to listen to basketball. Warriors. Next round.
Doesn't look good though. The Suns are getting destroyed when I last watched. It was halftime and they were trailing by 30. So I imagine Mavericks are coming in. I'd rather play the Mavericks than the Suns.
Are they playing now?
Yeah, the Suns were trailing by 30 at the half.
Game 7.
Game 7.
Are you serious?
Yeah. At At Phoenix. At Phoenix. They've lost, haven't they?
Mavericks won. Mavericks
I
won.
would rather face the Mavericks than the Suns, as if I were the Warriors.
Do you follow NBA?
Not really.
Oh, okay, no, so you wouldn't appreciate what much of an upset that this is Dallas beating the Suns over 7 games.
Did not expect that.
Crazy.
That is insane.
Doncic is a legend, honestly.
Yeah.
Wow.
I would rather face the Mavs though, just because I think we match up better. And I'm not I'm not feeling good because the Warriors, even though they won the last series, they didn't play particularly well.
Yeah. Game 6 Clay sort of saved them.
Yeah, and of course, Kevon Looney getting those 22 rebounds.
Wow, Mavericks beat the Suns. That's nuts.
Yeah, so... and I think whoever comes in the West is going to get smashed by Boston anyway.
Oh, yeah, Boston
beat the Bucks today.
Yeah, today. Oh, that was amazing. Like, in Boston, I thought Bucks would win the series, but Boston done well.
They'll get smashed by Boston.
Bucks
wins.
missed Middleton, though, so I think that's an excuse for them.
Yeah.
But, wow, Mavericks. Full of second rounders, plus Doncic, and then it's they managed to beat the Suns. That's crazy. So it's Miami versus Celtics.
Yeah,
Celtics gonna win that one, I think.
I would imagine.
Mavs
so.
versus Golden State.
That's anyone's game, but I'd imagine whoever comes in would lose to Boston. I think Boston's way too strong. The fact that they swept the KD and the Nets.
Oh yeah, but
they swept them.
It's true.
Swept.
True.
So that's something. I wouldn't have imagined KD ever getting swept.
I'm just wondering.
It's Kevin Durant, man. Come on.
They still
7 feet tall, hits shots anywhere. Like
but he had no support though.
He had Kyrie.
Kyrie's not a support. He I know. I think Kyrie's rubbish since the Cavs days. Like he's overrated.
He needs LeBron.
Oh no.
He needs LeBron to help him out.
I think he does. And anyway,
Let's see.
Not a big fan of Kyrie. Not a big fan of Kyrie for the negative influence he has in the for his teams that he's involved in, even with the Cavs. After they won their sort of titles. Anyway, it's a science podcast, not a basketball podcast. So
get super excited and deviated, but what right?
All right. So thanks for listening, and we'll come back to you with another episode in a few weeks' time. So until then, enjoy, and I'll see you around.
See you
We'll
guys.
see you around.
See you.
Yeah. Bye bye.